Hello students, welcome to today's class. I am Bernisha, lecturer from Emmanuel Aristide College of Pharmacy. Today we are going to see about the preparation method and evaluation of suppositories. In the last video, we have learned about what are suppositories, their advantages, disadvantages, along with what are the suppositories bases we are used in the preparation of suppositories. Today in this video, we are going to detailedly explain about learn about the preparation methods. What are the different types of methods which is used in the preparation or formulation of suppositories, and what are the methods mainly used for the evaluation of suppositories. First, the suppositories are the semi-solid dosage forms which is intended for the insertion into the body cavities such as rectum, vagina or urethra. In this body, it melts and softens. They are dissolved to produce as the local and systemic effects. There are various methods which is used for the preparation of suppositories. The preparation of suppositories mainly involves the incorporating the medicament into the suitable base, and we have to shape it into a desired form. Mainly, there will be have some type of bases and stability for ingredients. The proper preparation which ensures the uniform drug distribution, accurate dosing along with the good physical characteristics. So the method of preparation mainly depends on the factors such as nature of the drug, type of base, and the stability of ingredients. Within this, what are there are various methods which is used for the preparation of suppositories. It includes mainly hand rolling method, compression molding method, and fusion molding method. These are the three commonly used methods which is used for the preparation of suppositories. First, we are going to learn about the hand rolling methods. Mainly, it shows that hand rolling and shaping. It is one of the oldest and simplest method of suppository preparations. In this method, mainly what are the steps involved means first we have to make a mass. First, we have to make a plastic-like mass which is prepared by titrating the base along with the active ingredient in the mortar pestle. With the help of mortar pestle, we have to properly titrate the active ingredient along with the cocoa butter to form a plastic-like mass. Once the mass is prepared, we have to make it into a ball with the help of hands, and immediately place it on the pill type. First, we have to. It is it is normally it is like the preparation of chapati dough. Like we have to prepare a mass with the help of hand, and we have to kneaded mass in a roll between the two fingers and to make it in a rod shaped units. After we have to properly make it into a ball like structure, we have to knead it properly, and with the help of the two fingers we have to make it into a rod shaped units. The rods are then cut into a pieces, then one end is pointed with the help of spatula. So these are the common steps which is used in the hand rolling and shaping method. In this images, we have seen that the hand rolling methods. In the first picture, we see that we are preparing the rod shape with the help of fingers. And in the next picture, we see that first we have prepared a plastic-like dough. Next, the dough is placed on a pile type. Next, we have prepared a rod-shaped dough with the help of fingers. Next, we have to cut it into a smaller pieces for the preparation of the proper suppositories. So these are the similar steps, simple steps which is used in the preparation of suppositories by hand rolling methods. Next is advantage of this method. First, there is no need for heat the base like cocoa butter because mainly the temperature we taken that 86 to 90 degree Fahrenheit. Mainly the cocoa butter should melt at 35 degrees Celsius, below 35 degrees Celsius, so that the temperature should not be higher than 86 to 90 degree Fahrenheit. If this happens, you will have to be retemperate. Tempering is a type of heat treatment used to increase the toughness or recovery of crystalline form. Mainly, we should not this method does not need any heating processes. Next, it also has some limitations because the hand rolling requires some of the practice and skill as well. So these are the advantage and limitation of the hand rolling methods. Both these methods have both advantages as well as the limitations. Next method is compression molding or cold compression method. Compression molding is a method of preparing suppositories with a mixed mass of grated suppository base and medicaments which is forced into a special compression mold. Mainly we have to taking the suppository base. We have to grate that suppository base with the help of grater. Next, we have to mix it with the medicaments and force it to keep it into a compression mold. It follows the same procedure with that of the hand molding, except the base containing the drug is compressed into the suppositories using a machine. The basic requirement which is used for the preparation is mainly we have to request the capacity of mold first determined for the compressing the small amount of base into the dice. Next, when the ingredients are added, it is necessary to omit a portion of the suppository base based on the density factors of the active ingredient. Based on the density factor, we have to omit certain portion of the suppository base, and it helps for the compressing this machine which is used for the compression should be determined by compressing a small amount of base and weighing the finished suppositories. Next, what are the steps used in the compression of suppositories? First, the suppository mass is placed in a cylinder. First, we have to place the suppository mass in the cylinder. In this picture, we know that we have to prepare the mass which is denoted by the yellow color point. We have to keep the mass in that place. Next, the cylinder is closed and pressure is applied on one end by turning a wheel. Here we are using a piston. The mass is forced out at other end of the mold. When we are applying force at one end, in here we are prepared. We have to add the mass we have prepared. The dough we have prepared. Next, by applying the force by pressure by piston in the one end. Next the mass is forced out to the other end in the mold. Next, the other end there will be a mold with a narrow opening and a stop plate. The mold itself, the mass will enters into the mold and there it gets molded perfectly with the proper shaping, and after that there will be a movable end plate so that the back of the die is removed, and when the additional pressure is applied to the mass in the cylinder so that the formed suppositories are ejected through that mold with the removing the stop plate. So with the help of this picture we can easily identify the steps which is involved in the formulation of suppositories based on the compression method. Next is what are the advantages and disadvantages of this method. The advantages is it is a simple method which is skipping the task of hand rolling. We should not need somewhat pressure or skills which is needed for the hand rolling method. The suppositories which is formed by this compression method is more elegant than that of the hand rolled one. And it should avoid sedimentation of blessed suppository bases. It avoids the sedimentation of bases, and compression is especially suitable for making suppositories that contains the heat labile medicinal substances. Heat labile medicinal substances are also easily formulated by using this compression method. What are the disadvantages means in other way aqueous causing entrap means there will be a weight variation, and there will be an oxidation of both the drugs and bases. So these are the certain advantages and disadvantages of compression or cold compression molding. And the next method is molding from melt. Molding from melt. First, in this method, we have to melt the suppository base and the drug. First, we have to take the base and melt it properly. And next, we have to add the drug towards it, and it also gets completely dissolved in that melted base. So after that, it gets melted properly. We have to pour it into the suppository molds. When the mixture gets congealed, the suppositories are removed from the mold. It is while seeing the picture, we can identify that that is the mold. Here we have closed it into it, and after within the mold, we have to pour the melted suppository bases and the drug. Once the mixture gets congealed, we have to remove it from that mold. First, we have for the steps. First, we have to melt the base and cool it into a consistently thick cream, and we have to add the solid drug with that it and stir it in it until it fully dissolved and proceed it in the usual way. Usually after that we have to congeal the mixture. After it gets congealed, where the suppositories are removed from the mold. And what are the molds we are commonly used in the preparation of suppositories? The common used molds are made up of stainless steel, aluminum, brass or plastic. These are the certain substances which is used for the preparation of molds. Commonly we are using the stainless steel molds. First, these molds may be reusable and disposable. We have to easily reuse these molds and it also be disposable. And commercially, these available for the preparation of rectal and urethral suppositories, so that it can be produced individually as also when there is a large number of suppositories are involved. Next, the fusion method can be used with all types of suppositories. The suppositories are generally made from solid ingredients and the drugs which are measured by weight. When they are mixed, melted and poured into the suppository mold cavities, they occupy the volume of the mold cavities. Usually they undergoes the density factor for the preparation of suppositories. So common molds which are used for the preparation of suppositories mainly made up of stainless steel, aluminum, brass or plastics. The molds which are prepared should be easily disposable and reusable. It should be useful for the preparation of, mainly the molds are available for the preparation of rectal, vaginal and urethral suppositories. These molds can be constructed based on as it produces both the individual as well as the suppositories of large numbers with various sizes and shapes. Commercial suppository productions. There will be having an automated filling or volumetric dosing pumps which are used for the preparation of suppositories, which produces the suppositories automatically, which does not need any manual they do not need any manual instructions. They just first the strips of perforated shells are passed beneath the dosing pumps and filled successively, passed through the cooling chambers for the solidification, then sealed and then packaged. It is very easy preparation. Mainly it is used for the commercial suppository production, and there is a requirement of large number of suppositories. These automated filling volumetric dosing pumps are used for the preparation of suppositories. Here mainly, first we have to place the strips of perforated cells beneath the dosing pumps. They have filled successively, and after that they will move towards the cooling chambers where they get solidified and then sealed and packaged. Usually the quality control procedures were done for weight, volume, leakage or conductor. The main advantages for this types of preparation means the product efficiency is increased, so that the thousands and hundreds of suppositories can be prepared at a time. During this hand roll time, we have to made only up to hundreds of suppositories per day. But now, with the help of this commercial suppositories productions, we can easily produce at least thousands of suppositories in a time. Next, at least no chances of contamination, as we does not need uses the manual hands or like hands like that. So there will be the less chance of contaminations and it is very low cost. They does not need any skilled persons for the preparation of suppository. So this commercial suppository production is one of the very easiest methods for the preparation of suppositories in the large quantities. So there are four types of methods we have learned in the preparations of suppositories. First one is hand rolling method, and the next one is compression or cold compression method, and the third one is fusion or melt fusion method, and the fourth one is automated filling of molds by volumetric dosing pumps. These are the certain methods which is used for the preparation of or formulation of suppositories with a suppository basis. Next is displacement value. Displacement value is one of the important topic which is to be take part in the suppositories lesson, which is a very important questions. The displacement value is nothing but it is defined as the number of parts by weight of medicament that it displaces one part by the weight of base. Uh, is mainly first we have to take that it is defined as the number of parts by weight of medicament that displaces the one part of by weight of the base. The volume of suppositories from a particular mold will be constant, but the weight will vary because the density of the medicaments usually differed from the density of base. Hence, the density of medicament will affect the amount of base required for each suppositories. Because the base we are chosen for the each suppositories mold will be equal in shape, but the medicaments we are used for the preparation of certain suppositories will be different in the density of bases. So with the help of the density we can easily find out how much amount of base is required in the for each suppositories. With the help of this displacement value we can easily find out the number of parts by weight the medicament that displaces the part of bases in the suppositories mold, which is very important in identifying the amount of base which is required for the preparation of suppositories. The calculations, how we will calculate this displacement value. The displacement value is equal to d by a minus c. The displacement value formula is d by a minus c, where we will know that a is the there how much quantity of base is needed theoretically, not practically. It's theoretically how much quantity of base is needed, which is up to a gram. And next, B is total weight of suppositories. That means up to play base plus drug. We have taken totally, if we are taking that it's 2 gram of suppositories, it will be the total weight of suppositories practically. So B gram will be weight of suppositories, that includes base plus drug. Next is amount of base needed. C gram is equal to 100 minus x is equal to B into 100 minus x by 100, which is equal to C gram. C gram is equal to B into B is the total weight of suppositories. We are taken practically into 100 minus x. That is the amount of base needed. 100 minus x. We does not know how much amount of base is needed. That is B into 100 minus x divided by 100 is equal to the C gram. Then the amount of medicament present in suppositories is equal to we know that the total amount of it is equal to total weight of suppositories, which includes both the base and drug. So the amount of medicament present in the suppositories is equal to B into 100 Bx divided by 100, which is equal to D gram. The amount of base replaced by the D gram of drug is equal to so amount of base we have, which is needed. So which is A minus C. When theoretically we know how much amount of base, then the total amount of base we needed. So A minus C will give that amount of base get replaced. So displacement displacement value is equal to D by A minus C. So D is the amount of medicament present in the suppositories to the amount of base replaced by the drug. So the displacement value is the ratio of total amount of medicament present in the suppositories to the amount of base replaced by the D gram of drug. So how there is a problem, calculate the displacement value of zinc oxide in the theobroma oil suppositories, which contains 40% of zinc oxide prepared in 1 gram of mold. Weight of 8 suppositories is equal to 11.74 gram. Here weight of 8 suppositories which contains theobroma is equal to thus we have to take it theoretically. So 1 gram of mold. So 1 into 8 is equal to 8 gram. So weight of 8 suppositories is which contains 40% of zinc oxide is equal to, which is given as 11.74 gram. So amount of theobroma oil, which is the base present in 8 suppositories, is equal to we know that remaining 40 100 minus A minus x. So 100 minus x is equal to 100 minus 40 is equal to 60 by 100 into B, which is 11.74, which is equal to the way we will get the amount of theobromine oil present in the 8 suppositories. Next, amount of medicament present in 8 suppositories. Amount of medicament which is 40 percentage, 40 by 100 into 11.74, which we will get the amount of medicament. So the amount of base replaced by 4.96986 gram of drug is equal to, so 8 minus total A minus B. So which shows that A minus 7.044, which is equal to 0.956. The displacement value is equal to D by A minus C. This is also another problem which is used the displacement value for the calculation of preparation and dispensing of iodoform suppositories, which has iodoform of 0.9 gram, and which also has the cocoa butter sufficient quantity to make the suppositories of 8 suppositories of 2 gram each. With the help of the displacement value, we can easily find out the amount of base and the total amount of drug with the help and displacement value. With the help of that, we can easily find out the quantity of cocoa butter which is required for the preparation of iodoform suppositories. Next, we are going to learn about the test for suppositories or the evaluation test for suppositories. Evaluation is nothing but we are going to check the quality of the suppositories. We have to check whether the suppository is good for the formulation or consumption. With the help of certain tests, we will confirm that the suppositories are good to consume or it is ready for the marketing purposes. First is uniformity of weight. Uniformity of weight means we have to check whether the suppositories are in correct weight. The weight of the suppositories and the average weight should not deviate from the certain limits. Disintegration test means nothing but it is the test which explains that at what time does the suppositories get fully dissolved in the body fluids. It is a very important test to denote at what time does the suppositories get melted or completely dissolved in the body fluids to give the onset of actions. Next is content uniformity test. Content uniformity test means nothing but it should know whether the drug should get uniformly distributed over the bases. It should know that the bases and drug should be uniformly distributed and it is fully incorporated with each other. Next is melting point determination test. Melting point determination test means it denotes how does the suppositories melt at a uniform time? At what time does the suppositories get melts in the body temperature? How does it melts? And the next is general appearance test. General appearance test is by checking their appearances as well as the color. It also evaluated the fissuring, exudation, pitting as well as the certain general appearances of the appearance of the suppositories by examining it visually. And the next is assay of active contents. Assay means we have to check the purity of the active ingredient which is used in the formulation of suppositories. And the next is liquefaction or softening time test for rectal suppositories. We know that the rectal suppositories should soften or liquefy at a certain body temperature. So we have to use certain apparatus to check the softening time or liquefaction time of the rectal suppositories, which is very important for the evaluation of rectal suppositories. Next is breaking test. Breaking test means we have to check how much pressure or how much mechanical strength does the suppositories would have, or it how much times which it can withstand the how much time how much strength which the suppositories have, and how will withstand the handling, packaging and storaging conditions. Next one is dissolution test. Dissolution test means nothing but the amount of drug that is distributed or dissolved at a certain time interval. We have to check the amount of drug that gets dissolved in the body fluids. So these are the 9 evaluation tests for the suppositories. They are uniformity of weight, disintegration test, content uniformity test, melting point determination test, general appearance test, assay of active contents, liquefaction or softening time test for rectal suppositories, breaking test and dissolution test. These are the 9 tests. We will briefly discuss this in this slides. Here the first test is uniformity of weight. Here for this test, first we have to weigh 20 suppositories individually. Then we have to determine their average weight. First we have to weigh the 20 suppositories individually. In order to find the average weight, we have to add the individual weights and we have to divide it by 20. Within that we can easily determine the average weight of suppositories. Here not more than 2 of the individual suppositories should deviate from the average weight. It should not deviate from the average weight and it should be not more than 5% and none deviates from more than 10%. So here any 2 of the individual weight should not deviate from the average weight with 5% within 5%, and in any one of the suppositories will not deviate 10% from that of the average weight. For the uniformity of weight, we have to weigh 20 suppositories individually. Then we have to determine their average weight. Then we have to check it with the limits in the monograph. Next is disintegration test. Disintegration test means we know that the larger particles will break down into the smaller particles and completely dissolve into the body fluids. First, for that we have to we have to know the apparatus. In that apparatus, there will be a transparent glass or plastic container with a height of 60 millimeter with an internal diameter of 52 millimeter. A metal device consisting of two stainless discs, each with a contain of 39 holes, each of 4 millimeter in diameter. Mainly, it is the apparatus. It is the disintegration test apparatus. Constructions. So the diameter of the disc is similarly to that of the internal diameter of the sleeve. The discs are separated by a distance of up to 30 millimeter. Usually a metal device is attached to that of the 3 equally spaced hooks. This is the disintegration test apparatus. Next, the method. First we have to place a suppositories on the lower perforated disc of a metal device. That the device is inserted into the cylinder which is attached into the sleeve. They repeat the same operation for further 2 suppositories, metal device and sleeve. Place each piece of apparatus in a vessel containing at least 5 liters of water at 37 degrees Celsius fitted with the slow stirrer, and by holding on the top of apparatus of which is 95 millimeter below the surface of water. We have to place the suppositories on the lower perforated disc. We have to repeat the same operation for the 2 suppositories. We have to take the apparatus is placed which is 90 millimeter from the top of the apparatus, and the below that it contains the 5 liters of water with 37 degrees Celsius, and which has the lower which with the slow stirrer. And the disintegration is complete when the molded suppositories are completely dissolved. When we have to note the timing up to when the molded suppositories get completely dissolved. The disintegration occurs in not more than 30 minutes for fat-based suppositories, and for water-soluble suppositories, disintegration occurs in not more than 60 minutes. Disintegration test is nothing but we have to check at how much time does the suppository gets completely dissolved in the body fluids. So when taking the suppositories in a disc which is a lower perforated disc, and in the above we have to make a disc which helps without moving of the suppositories. Then we have to put the suppositories in the water which has slow stirrer, and the water will be around 37 degrees Celsius. We have to hold the apparatus which is 90 millimeter just below the 90 millimeter below the surface of the water. Next, the disintegration is complete. When we have know that when the disintegration is completed by knowing that suppositories were completely dissolved in the water. For fat suppositories, it may occurs more than 30 minutes, not more than 30 minutes. For water soluble suppositories, the disintegration occurs will be not more than 60 minutes. Next is content uniformity test. For content uniformity test, we have to take 10 suppositories to determine the active ingredient of the each suppositories by using a suitable analytical method, which will be not more than one of the individual values. The subtract is outside the limit, that is the average value is none of them is outside the limit of 25% of the average value. First, we have to take this 10 suppositories. Here, the active ingredient of the each 10 suppositories, we have to check it by the suitable analytical method, which is the average value should not be more than that of the 25% of the average value. Next is melting point determination test. Melting point determination test means in this test, the suppositories, we know that the suppositories melt at the body temperature. So the melting of the suppositories rapidly at a temperature not more than 10 degrees Celsius above the point of complete fusions. Here first we have to insert one end of the glass capillary tube into the melted substance. So the column of the substance become 8 to 12 high, rise into it. Cool the tube up to 15 degrees Celsius and maintain the temperature at 15 to 17 degrees Celsius and not less than 16 hours. Attach the tube to the thermometer in the heating vessel containing the water at 15 degrees Celsius so that the lower end of the column of the substance is 30 mm below the surface of the water. Heat the water with a constant stirring so that the temperature rises at the rate of 2 degrees Celsius per minute. The temperature at which partially melted the substances begin to rise in the tube is regarded as the melting point. The melting point of suppository should not rise more than that of the given in the monograph. So we have a monograph which shows the certain melting point range when we are dipping the suppositories under 15 under the in a column which has 15 to 17 degrees Celsius temperature. We have to place a thermometer in that column and we have to keep the substances 30 mm below the surface of the water with the constant heat and stirring so that the temperature rises at 2 degrees Celsius per minute. So the first minute the temperature will be 15 degrees and the next minute it will be 17 degrees Celsius. So that the per minute the temperature will increases 2 degrees Celsius per minute. When the temperature reaches the certain amount, when the substances partially melted and begins to rise in the tube, it is recorded as the melting temperature of that suppositories. There will be the certain melting point range in the monograph. It should not exceed from that of the limit from the monograph. Next is general appearance. General appearance is nothing but the suppositories. We are checking the quality of the suppositories, thereby looking the outer appearances. The suppositories, when cut longitudinally and examined with the naked eye, there the internal and external surface of the suppository should be uniform in appearance. When we are cutting the suppositories longitudinally, we have to examine it with the naked eye so that the internal and external surfaces should be uniform in appearance. The compliance with the standard indicates satisfactory subdivision and dispersion of suspended material. While we are examine with the naked eye, we know that there will be a proper dispersion, uniform dispersion of the active ingredient with that of the bases, and the suppositories should be uniform in both internally and externally. The surface appearance and color can be verified usually to assess the absence of fissuring, absence of pitting, absence of exudation, absence of migration of active ingredients. First, we have to check the appearance, color. Mainly, we have to indicate we have to verify the suppositories should be absence of pitting, absence of exudation, absence of migration of active ingredients. Here, pitting means nothing but there will be some holes in the suppositories after demolding it from the mold. Exudation means there will be the oozing of liquids when we are cutting it in the longitudinally. So we have to check whether the suppositories should be absence of pitting, absence of exudations, and absence of fissuring. And we can easily found out there will be a non-mixing of uniform distribution of base and the active ingredients, so that the active ingredients gets migrated from that inside the suppositories. So in the general appearance, with the help of the naked eye, we should examine the fissuring, pitting, exudation, and migration of the active ingredients. This is the general appearance qualities check of suppositories. And the next is assay of active contents. Before getting it into the marketing sites, we have to check the assay of active contents present in the suppositories. The official limit for the active contents is around 95 to 105 degree, 5 percentage of purity. For example, mainly of it's for glycerol suppositories. First, we have to dissolve the number of suppositories, which is equal to 8 grams of glycerol in the 50 ml of water, and add a quantity sufficient to produce this into 250 ml of water. From that, we have to take up to 5 ml of the solutions and 150 of the ml of water, and 0.25 ml of bromocresol purple solution is added to it. So we have to add 0.1 m of NaOH to neutralize the blue color of that indicator. Then we have to add 1.6 gram of sodium metaphenolate and allow to stand for 15 minutes, and we have to titrate it with 0.1 m NaOH to the same blue color. Each ml of 0.1 m NaOH is equal to 0.00921 grams of glycerol. So that it is mainly we have to check the purity of the active ingredient which is present in the suppositories. For that, first we have to take up to 8 gram of suppositories which contains, which we have to dissolve the suppositories which contains 8 gram of glycerol in a 50 ml of water. Then we have to make up it into 250 ml of water. Then we have to take 5 ml of solution. Then we have to take 5 ml from that solution. To that we have to add the 250 ml water and 0.25 ml of bromocresol purple solution, which act as the indicator. In order to neutralize it, we have to add 0.1 m NaOH. Usually we have to titrate this with the 0.1 m NaOH to get to the blue color end point, and after that we have to add 1.6 gram of sodium metaphenolate phenolate and stand for 15 minutes. After adding the bromocresol purple solution, we have to add 1.6 gram of sodium metaphenolate and we have to stand it for 15 minutes. After that, we have to titrate it with 0.1 m of NaOH so that we will get the blue color solutions. Here, each ml of 0.1 m NaOH is equal to 0.00921 gram of glycerol. Next is liquefaction test for the rectal suppositories. Liquefaction is nothing but here usually the suppositories get melted at the rectum temperature. So it is the modification method which is developed by the Krosansky, which is another useful test for the finished suppositories. First, we have to it consists of U-tube partially submerged in the constant temperature water bath. First, we have to immerse the U-shaped tube into the constant temperature water bath. A construction of one side hold the suppository in place in the tube. A glass rod is placed on the top of the suppositories. At the time for the rod to pass through the construction is recorded as the softening time, which denotes the time. Softening time means when we are inserting the suppositories into the rectum, within how much time does the suppositories get melted or get softened. That should be noted. Here, when it is placed in the U-tube above the suppositories, there we can place the glass rod. When we are placing the U-tube under the submerged constant temperature water tube, immersed into the water tube, with the help of the temperature, the suppositories get melted slowly, so that when it's melted, the glass rod get moved through the constructions. We have to note the time. That time period is known as the softening time. This can be carried out in the various temperature by changing the temperature of water bath. As a quality control check, which can be also studied to measure the physical stability over time. So that when the lower temperature, it should be stable, solidify, and when it reaches the 35 to 37 degrees Celsius, it gets melted or softened. So this is the liquefaction test for the rectal suppositories, or the test for the softening time. So breaking test is another evaluation test for the suppositories. It is also known as the hardness test. It is mainly used to determine the mechanical strength of the suppositories to ensure they can withstand the handling, packaging, and transportation without any breaking. The major principle is when the suppository is subjected to the increasing weight or pressure until it breaks, the force required to break it indicates its strength. Next, for that, first we have to take a suppository sample, usually 1 to 3 units. We have to place the suppository horizontal between the two supports of the apparatus. We have to gradually increasing the weight of force on the suppositories. We have to continue add the weight on the at a regular intervals, so that at 1 minute intervals 200 gram of weights are added. Mainly this test is conducted by placing 600 gram on the platform. First we have to keep 600 gram on the platform. At 1 minute intervals 200 gram weights are added to it, and at the weight which is suppository collapses is the breaking point. So we have to determine, note the minimum weight required to break the suppositories. The repeat the test for the other samples, and we have to calculate the average breaking strength of the suppositories. First, we have to keep the suppositories between the two supports of the apparatus. In one side, we have to add the weight. Usually, at the starting time, we will keep up to 600 gram of on the platform. At 1 minute interval, we have to add 200 gram of two weights are added, and at which weight the suppositories get collapses is known as the breaking point of the suppositories. Usually, the suppository this test is used to ensure that the suppositories can withstand handling, packaging, transportation without any breaking. This test is carried out for the different samples of suppositories and the different shaped suppositories to withstand the breaking forces which is useful for the handling of production, packaging, etc. Next is a test is dissolution testing. In this, the suppository is placed in a dissolution medium which is maintained at our body temperature, and here the amount of drug which is released over time is measured. Here first we have to fill the dissolution vessel with a suitable medium. Usually we will fill it with a phosphate buffer pH around 7.4. Next, we have to maintain the temperature of the water bath around 37 to plus or minus 0.5 degree Celsius. We have to place the suppository in the basket or the holder to avoid the floating of the suppositories. We have to rotate the apparatus at a specified speed of 550 to 100 rpm. Next, at a regular intervals, we have to withdraw the samples of the medium so that and replaces it with a fresh medium to maintain the constant value. We have to analyze the samples using the UV spectrophotometer or a suitable method. We have from that we can easily find out the amount of drugs which is released over the certain period of time. Here the plot of drug release versus time is calculated, and we can easily found out the percentage of drug release at different time intervals. With that we can easily find out the drugs which is released at the drug release of the suppositories. This is the dissolution testing. Okay students, in this video we have learned about what are the preparation methods which is used for the preparation and formulation of suppositories, which includes hand rolling method, compression or hold compression method, and fusion or melted method, and automated dosing pump method. These are the certain methods which is used for the formulation of suppositories. And the next we have learned about the displacement value, which is very important for factors for the formulation of suppositories. Next, we have learned about the evaluation tests which is involved in the quality checking of the suppositories or finished formulation of suppositories. In this evaluation test, we have learned about the uniform weight variation test, content uniformity test, dissolution test, disintegration test, liquefaction or softening time test of the suppositories, hardness test, breaking test. These are the certain tests we have learned in that evaluation test for suppositories. These evaluation tests were very important for the quality, important to check the quality of the suppositories. Okay, students, I hope you all have learned very nicely about the evaluation and the preparation of the suppositories. Let's see in the next video. Thank you.